Morning, everyone. I'm Mara Becker, and on behalf of the Pediatric Trials Network and the NICHD, I'd like to welcome you to our fall virtual meeting on developing clinical pharmacokinetics and safety trials in Down syndrome. As many of you know, we thought we were going to have this meeting in person this summer with the Down Syndrome Congress and the DSMIC meeting, but as COVID would have it, we are all virtual. So thank you so much for joining. I'm hoping that you find this morning to be a value add, and we really look forward to your participation. So the meeting goals are at the top of your agenda, but I'd like to review them because we have a lot that we'd like to accomplish today. Primarily, we'd like to increase the dialogue and collaboration between all these great key stakeholders. There's so many people from very different domains that are attending today, and we're really thrilled that you took the time to do so. And we all have a very shared goal of improving clinical outcomes in the population of patients with Down syndrome. We'd like to raise awareness of the therapeutic challenges faced by children with Down syndrome and to develop educational and research strategies that include these individuals with Down syndrome as contributors as well as participants. We want to discuss the challenges and lessons learned that we've learned along the way in this first year, especially as we've been starting to get involved in the POP02 study, which we'll talk about briefly. And lastly, and importantly, we'd like to solicit your input on a prospective randomized clinical trial that's currently in development. So the Pediatric Trial Network Down Syndrome Project has an overarching goal, and that's to create a pediatric trial infrastructure under which on and off patent therapeutics can be studied to improve the care of children with Down syndrome. And we'd like to do this via two different mechanisms. The first is through the development of a pediatric clinical trial network for patients with Down syndrome under the umbrella of the PTN. The second is through development of a bidirectional training program for clinical researchers. So within that clinical trial network, we have two mechanisms in which to obtain that goal or to achieve that goal. The first is through involvement in the POP02 study, which stands for Pharmacokinetics, Pharmacodynamics, and Safety Profile of Understudied Drugs Administered to Children per Standard of Care. Hence why we call it POP02. It's a lot easier. We're going to use POP02 as a platform to train a core set of Down syndrome sites on clinical pharmacology-based research, to learn early from those core sites what are challenges unique to patients with Down syndrome and the clinicians that treat them, to build relationships with these thought leaders and site staff, and to collect pharmacokinetic and pharmacodynamic or PK/PD data in real-world setting in children with Down syndrome. So this study evaluates the pharmacokinetics of understudied drugs that are currently being used in children as standard of care. And so for this cohort of sites that treat children with Down syndrome, the drugs of interest that we chose include guanfacine, dextroamphetamine, amphetamine, sertraline, and risperidone. The second aspect of this will be developing a prospective therapeutic trial assessing the safety and efficacy in children with Down syndrome. And so we'll be discussing that later on in the day. The second component is through what I prefer to use as multi-directional trainings. And we'd like to create trainings that are geared specifically towards the PTN investigators who don't have much experience working with individuals with Down syndrome and their families, as well as the clinicians who treat individuals with Down syndrome who may have less experience in the conduct of clinical pharmacology-based trials. We want to utilize the lessons learned from POP02 to inform trainings and to prepare those new sites for this prospective trial that we're trying to develop currently in individuals with Down syndrome. But most importantly, we want to capitalize on these relationships that we're trying to build with these stakeholders to further research in this domain. So let's talk about some meeting housekeeping. Please, during each session, please use the chat box to ask questions during the sessions. And if you have a question that's directed to a specific speaker, please specify in your chat. We'll have the speakers available during their sessions to answer chats along the way. There'll be a moderator, yours truly, who will ask a few questions live streaming at the end of each general session with the speakers from the session. And if there are remaining questions that we haven't gotten to during that session or through the chat, we will gladly respond to those questions and circulate them via email after the meeting. So I'd just like to send a special thanks out to all the people who made this meeting possible, including our Down syndrome thought leader group, the Include leadership, NICHD leadership, the PTN steering committee and leadership, and the DCRI staff who've been working so hard to pull this off. I'd like to thank the MS Corporation for their investment in this work, patients and families who are attending and volunteering as part of this meeting, and our guest speakers and collaborators who've been along the ride with us and informing and some of this work along the way as well. And last, of course, I'd like to thank GlobalCast MD who are making us all look good today. And I appreciate everybody's efforts in pulling this day off. I hope you enjoy the meeting. Thank you.
Hello, I'm Kenesha Zimmerman. I am the chair of the Pediatric Trials Network Steering Committee, and I'm very pleased to be here today to talk to you about the Pediatric Trials Network. Here are my disclosures. So what is the Pediatric Trials Network? It was designed to create an infrastructure for investigators to conduct trials that improve pediatric labeling and child health. We've been sponsored by the NICHD, and our success is defined by improving dosing, safety, labeling, and ultimately child health. And our focus is on off-patent therapeutics. The organizational structure of the Pediatric Trials Network is as follows. I know it looks a bit complicated, but the point here is that we have a number of people who are involved in a number of organizations, and our goal really is to be very collaborative. We have multiple different areas in which we are focusing, including pharmacometrics, safety, ethics, and community and stakeholder engagement, devices in particular, clinical pharmacology, and then site training and investigator mentorship. This effort is led by Dr. Danny Benjamin, who is the PI of the PTN at Duke. And as I mentioned, I'm the chair of the steering committee. So the PTN has a very unique structure. The NIH develops a priority list of off-patent therapeutics, which you can see the link here. Investigators submit a study concept to PTN, and then the administrative core within PTN reviews the science and feasibility. After that concept sheet has gone through the administrative core, the PTN steering committee then reviews that concept for science and feasibility. And if approved, the PTN performs protocol development team. They develop a protocol chair, thought leaders, pharmacologists, operations expert, everyone who's needed in order to get this moving. The PTN then selects sites based on site study interest, availability, and a previous history of enrollment, and then we execute the clinical trial. We've been successful using this model. We've studied more than 18 therapeutic areas, enrolled more than 11,000 participants, and submitted 26 CSRs to the FDA. We've successfully changed 15, now 16 labels with information about children within those pediatric labels. And this is all since 2010 when this award was first given to Duke University. So why is the PTN successful? I think that comes with recognition of the current challenges in the pediatric clinical trials. There are many, as you know. Recognition and then finding ways to address them, which is what we're going to talk about over the next couple of slides. We all know that many of the clinical trials that attempt to be done in pediatrics are limited by the limited number of patients, the blood volume, perceived study risk, variability in site enrollment and contracting, competing research priorities, low consent rates, validated endpoints, research funding, trained investigators, all of these things you can see here that create a potential problem for making sure that we get pediatric clinical trials done and done in a timely manner and done with the best quality evidence there. So many pediatric trials usually have requirements for large sample sizes. There are extensive inclusion and exclusion criteria, and we recognize this as a potential issue and barrier to actually enrolling children. What we've found is that being very selective leads to lots of difficulties in enrollment, but we want to make sure that our populations are actually reflecting the populations who are going to use the drugs. And so we have inclusion and exclusion criteria as opposed to that large list that you saw there. We have narrowed, this is an example of what we narrowed down to the inclusion and exclusion criteria, just 6 things here. And the way that we're able to do this is because we have very close relationships and interaction with the FDA to try to tell them about what's feasible and what we can do with the information that we will be collecting. This makes it easier for sites, easier for patients, easier for parents and everyone. And so we've been very successful in doing these things. Another challenge is limited blood volume. You can see babies in particular have a lot lower blood volume than the adult. And this becomes an issue. This becomes a situation where people are very worried. So we have done things like develop sensitive drug assays and minimal sampling methods in order to make this better. Things like dried blood spots, multiplex assays so it can test multiple drugs at one time, sparse sampling, and then using population pharmacokinetics. Through the Pediatric Trials Network, this has been made possible by collecting few samples from lots of patients in their everyday clinical care, and then being able to translate that using population pharmacokinetics to understand the disposition and of drugs within kids of all different shapes and sizes and ages and different clinical scenarios. This is made possible by our DCRI Pharmacometric Center, led by Dr. Cohen Wolkowitz, as well as Christoph Hornick and Stephen Balovic. We have a number of people who are supporting this effort, and it allows us to really be focused in doing clinical pharmacology. Another challenge that we see is site variability and prolonged time to establish site contracts. One of the things that the PTN recognized early was that master protocols were going to be necessary. Master protocols where we're thinking about how we might be able to enroll kids in multiple drugs, so you're not having to contract over and over and over again. And that's one of the things that we're doing with the Include network here. We have one of the studies, as you've mentioned, as you've heard about before, the pharmacokinetics of understudied drugs administered to children per standard of care, which studies more than 50 drugs at a time. It's enrolled more than 3,000 patients. We have more than 40,000 enrollments per month, and we look at a number of different special populations, collecting them in their place of care. It's really the ultimate pharmacoepidemiologic experiment. Another challenge is competing research priorities. You may be well aware that often one NICU or one center is approached with multiple studies to do, and people are overwhelmed by what they can do. Co-enrollment largely has been problematic for a lot of these studies. However, we've approached this from multiple things. One, we allow co-enrollment. We want kids to be enrolled as long as we're not exceeding things that are dangerous, like the amount of blood that we're taking from kids. We also have learned to develop protocols that complement and fill the gaps. Number of our protocols have been focused on things like neonatal antimicrobial studies, PK studies, and special populations where other people really aren't touching. As I mentioned, we also allow for co-enrollment, and we discuss co-enrollment with potential site investigators during the site selection process so that we're all on the same page. We're even willing to talk to investigators from that are leading the other studies in order to make sure that we all want to be in this together and we all want to move pediatric drug development forward. Another challenge, as we know, is low consent rates. Clinicians are really concerned about parental burden. Parents are really concerned about study invasiveness, unclear personal or child benefit. And part of our effort in order to solve this issue is to make sure that we have parent and participant engagement that is helping us to understand what their contributions are to the study and how they really sacrifice. Also, making sure that we're thanking them and providing them feedback as to what's happening, and getting their input on the study protocol so that we know that we're doing things that are reasonable and that people will see as valuable. And then finally, the lack of pediatric clinical pharmacology expertise has really been a limitation in being able to do pediatric drug development and enroll kids. We have tried to solve this problem through a number of venues. One is multiple T32 programs that are focused specifically on clinical pharmacology. We collaborate with the UNC Eshelman School of Pharmacy. We have multiple fellows, multiple alumni who've focused specifically on this. And we do a lot of data sharing within PTN to make sure that we are collaborating and learning from ourselves and learning from other people. We also know that partnerships are really important. It's one of the reasons that we're really excited about the partnership with Include. We've done this model before with the Idea States. They've partnered with NPOPs and been able to help them understand how to do clinical research in a regulatory compliant manner. We also are looking to the future. We have an R25 program that's very closely linked to the Pediatric Trials Network with high school students and college students and educators who come to the Duke Clinical Research Institute every year. They learn research methods and ethics and have a hands-on clinical research experience. So the PTN has multiple lessons that have been learned throughout our time. We know that being collaborative is really important. We know that engaging stakeholders is really important, and we've done that. We've been able to maximize the protocol efficiency with simple designs and standard of care procedures and making sure we're inclusive. And we've been maximizing operational efficiency with master contracts, careful site selection, and then some template documents when appropriate. I will stop there and I'm happy to take any questions.
Good morning, and thank you for allowing me to present on the Include project. My name is Melissa Parisi. I'm the chief of the Intellectual and Developmental Disabilities Branch at the Eunice Kennedy Shriver National Institute of Child Health and Human Development at the NIH. I have no disclosures. Some of you may have seen the announcement in early October about the NIH awarding $60 million in Include funding for Down syndrome research in fiscal year 2020. This is a snapshot of the post that was promulgated by my director, Dr. Diana Bianchi, on her director's blog, and provided some background about this initiative and what we're doing in Include. We really hope that Include is going to provide an opportunity for us to move closer to personalized medicine for families and children with Down syndrome, and something that will really improve their quality of life. Before I define Include, I wanted to give you a little background as to the basis for how we got here and what was the motivation for creating this initiative. There have been 3 congressional directives that have really helped define our role at NIH with regard to Down syndrome research. In 2018, we were directed to develop a new trans-NIH initiative to study Down syndrome, in particular to yield new scientific discoveries that would improve the health and neurodevelopment of individuals with Down syndrome, as well as typical individuals who are also at risk for many of the same co-occurring conditions, such as Alzheimer's disease, leukemia, heart defects, immune system problems, autism, and others. In FY 2019, we were directed to continue implementing the Include initiative and also promote the development and funding of early stage investigators to expand the current pipeline of Down syndrome researchers. And in this fiscal year, we were directed to continue to provide grant opportunities and expand the pipeline of researchers in this field. As a result, we developed the Include initiative, which stands for Investigation of Co-occurring Conditions Across the Lifespan to Understand Down Syndrome. As you know, at NIH, we really like our acronyms, and this one is a classic. The Down Syndrome Initiative Include has 3 different components. The first, which is represented in this Venn diagram as the lavender circle, is really focused on targeted high-risk, high-reward basic science studies on chromosome 21. The second component is focusing on cohort development and is represented by the salmon-colored circle. This goal is to really develop a large study population of individuals with Down syndrome across the lifespan. And the third component, represented as the green circle, is focusing on inclusion of individuals with Down syndrome in existing and future clinical trials. So when we talk about a trans NIH initiative, we mean that this is a project that is involving 18 different institutes and centers across NIH, and they are covering all the major co-occurring conditions in Down syndrome. We're referring to vision problems, cardiovascular, intestinal, pulmonary issues, developmental, ADHD, behavioral issues, Alzheimer's disease, immune system problems, and many others. And by including these different institutes, we have a better chance of actually covering all of the major co-occurring conditions for people with Down syndrome. So Include has had a significant impact on Down syndrome research at NIH. In the early 2000s, we were hovering around $20 million a year total investment in Down syndrome research. That started to pop up in 2015, 2016, but really in 2018, with Include funding of $23 million, an additional $35 million in 2019, and then finally $60 million this year, we're now at an estimated $113 million of total Down syndrome related research investment across NIH. This is a staggering increase and really represents the success and the investment and the commitment of NIH in Down syndrome research. How have we done this? Through a variety of funding opportunity announcements. We issued 13 FOAs to further expand and develop the Include project in 2020. 3 of them were building off of 2019 initiatives. 1 was looking at basic science awards, but 2 of them were really focused on that third component in clinical trials. 1 is a clinical trial readiness R21, and 1 is looking at clinical trials, specifically 2 phased awards that are R61/R33s, 5-year awards to promote clinical trials in Down syndrome. In 2020, we also had 2 RFAs to help develop the cohorts, and in particular, funding for a data coordinating center, as well as RO3s for data analysis and secondary data analysis. We also issued 8 new notices in this past year to help promote development and training for fellowship and career development awards, to help develop animal models, and to also continue to support supplements, R01s, and competitive revisions, specifically around COVID-19. Many of these FOAs continue to have due dates into fiscal year 21 and 22, so these are still open and accepting applications in the next couple years. With regard to COVID-19, representing the unique susceptibility potential for people with Down syndrome to develop complications of COVID-19, we've issued an urgent competitive revision and have several upcoming due dates for this particular supplement proposals. This slide represents a snapshot of what we have funded in this 2020 year with regard to Include research. On the left, the pie chart shows each of the institutes at NIH that is managing awards that are issued under Include, with about 19 million going to aging, 16 million going to human development, my institute, about 9 million going to heart, lung, and blood, and then cancer, arthritis, immunology, and others receiving a certain amount of funding as well for Include related projects. The $60 million is also distributed across the three components of Include, again with basic science, clinical cohort, and in green, the clinical trial aspect of those awards. And keep in mind that this is a total of 42 different awards that have been funded in this past fiscal year. Some of the awards that have been funded include those in the basic science domain, which include trying to understand the role of genes on chromosome 21 in skeletal development, cognition, cellular processes, immune function, and even risk factors for heart defects in Down syndrome. I've already mentioned the commitment to develop a data coordinating center for Include that has been funded this year. And we're also looking at trying to understand leukemia, celiac disease, autism, and other conditions in Down syndrome, as well as strategies to recruit underrepresented minorities into Alzheimer's disease research related to Down syndrome. And finally, under clinical trials, we are funding an R01 to test whether a hypoglossal nerve stimulator can improve language development in those with obstructive sleep apnea. In the training domain, we are supporting 24 new junior trainees and researchers. And all of the awards that have been made under Include are listed on the NIH Include website that is at the bottom of this slide. Given that we're focusing today on clinical trials, I wanted to highlight the 6 clinical trials that have now been funded under Include. The first 5 were funded last year, so they're just completing the first year of their R61 phase. One of them is looking at clinical trials to prevent Alzheimer's disease and Down syndrome. One is looking at JAK inhibition in autoimmune skin disorders in Down syndrome. The next 2 are focusing on obstructive sleep apnea, with one looking at positive airway pressure and compliance with regimens to promote the use of PAP for individuals, and particular children, with obstructive sleep apnea. Another one is looking at medications that can be used to treat OSA to improve cognition in children with Down syndrome. The 5th award is really developing an assessment and medication treatment for ADHD in children with Down syndrome, with a focus on stimulant medication. And this is led by Drs. Anna Espenson and Tanya Froelich at the University of Cincinnati. And then finally, the clinical trial that I just mentioned that was just awarded about a month ago to look at the effects of hypoglossal nerve stimulation. And this is led by Drs. Christopher Hartnick and Brian Scottco at Massachusetts Eye and Ear Infirmary. So Include does not work in a vacuum, and we're very excited that Include is now partnering with the Pediatric Trials Network to really help support drug studies in children with Down syndrome. So one of the ways in which we're doing this is through the opportunistic studies in children already receiving medications who have Down syndrome being conducted across the PTN network. Another goal is to help train investigators to conduct research involving children with Down syndrome. And finally, what we'll be talking about today is looking at the upcoming clinical trial of guanfacine for treatment of ADHD in children with Down syndrome. The Include project also has a number of upcoming events. We have more funding opportunities under development in addition to those that are being continued from this current year and this past year. We have clinical trials support and training for investigators that we're very interested in supporting. We are looking forward to establishing the new Data Coordinating Center for Include, and we have a virtual workshop that is scheduled for next week on the intersection of basic science and clinical cohort development. The URL to register for that meeting is available, and a draft agenda is also posted on the website. So with that, I'd also like to close with just an announcement about the Include research plans that are undergoing an update and revision. Our goal is to publish the revised research plan in 2021. The responses to a request for information are posted on the Include website, and we plan to post the draft new research plan by the end of this year. So look for that, and please be prepared to give us helpful feedback on that. With that, I'd like to thank you for your attention and look forward to the rest of today's meeting. Thank you.
Good morning, everyone. And I would really like to thank the PTN for this opportunity for me to speak on the clinical overview in terms of clinical variability and therapeutic challenges in individuals with Down syndrome. My name is Priya Kishnani. I'm Division Chief Medical Genetics at Duke University Medical Center. I have no disclosures. I think just to set the stage, it's important for us to recognize that Down syndrome is the most common chromosome condition, 1 in 792 live births, and there are about 206,000 individuals with Down syndrome in the United States alone. Majority have intellectual disability, which is in the mild to moderate range. The IQ range of individuals with Down syndrome is between 30 to 70, but with a mean of 50. And then there's another concept of Down syndrome and aging. It's hard for us to recall that even in 1929, the mean age for an individual with Down syndrome was 9 years. And today, our current estimate is nearly 60 years. So a lot has been accomplished through the decades in terms of helping our understanding of individuals with Down syndrome in terms of survivability and quality of life. But yet there are many, many challenges and considerations which we must keep in mind. The one important thing is that chromosome 21 contains about 225 genes, and there are certain regions which are called critical regions, and these have been estimated by studying families with a partial trisomy. But again, the point to make here is that with 225 genes and their interactions with each other, as well as with other genes in the system, it's hard to really get a full handle of how an individual with Down syndrome will present. And this comes to the next point of the clinical variability that we see in individuals with Down syndrome. So there are certain features which are common in terms of facial features, but when we start breaking this down further, there's a widely varying range in terms of degrees of intellectual disability. And in the study, looking at 121 individuals with Down syndrome with a mean age of 31 years, you can see that some were in the category of mild, some in moderate, some with severe, and then about 20% with profound disability. It also is important to know that higher levels of cognitive abilities typically correlate positively with better speech, and that the severity is correlated with increasing age, with tooth loss, with visual and hearing impairment, and when they have other comorbidities like epilepsy and or stroke. In addition, individuals with Down syndrome also have sensory impairments with varying degrees of visual impairment. And these hearing loss could be both sensory neural as well as conductive. And this then results in poor speech production. So now let's again look at another aspect, that the variability in presentation that can impact cognition can further be modified because of the underlying clinical aspects in an individual with Down syndrome. For instance, the presence or absence of congenital heart disease, thyroid disease, celiac disease, diabetes, depression, Alzheimer's disease, and then again, certain central nervous system involvements that we see in individuals with Down syndrome. a key feature for, I think, us to learn as a group or recognize is that individuals with Down syndrome do have language deficits, but when you match it on mental age, individuals with Down syndrome exhibit a significant delay in expressive language skills when you compare it to those with other developmental disabilities, and that the language production is lagging behind language comprehension. And thus, again, expressive language deficit is a major concern. And the greatest delays here is in syntax and in phonological processing, like using sounds to process language. So when we see individuals with Down syndrome in the clinic, it's very interesting to know that some are very high functioning with being extremely verbal and some who have very limited speech. Additional considerations, earlier onset of Alzheimer's disease in individuals with Down syndrome, but how early is early and how often are we delaying the diagnosis or misdiagnosing or delaying treatment initiation? This occurs quite often, but once again, to remember that the prevalence is up to 55% in individuals with Down syndrome in the age range between 60 to 69 years. Often we miscall a diagnosis of Alzheimer's disease, which could just be due to hypothyroidism or significant depression. So these all need to be taken as one bucket in terms of this variability in clinical spectrum. Now let's talk about the other variation, and this is in drug metabolism. We cannot have really a trickle-down effect of what we learn from other individuals because there could be differences in pharmacokinetics, pharmacodynamics compared to typically developing children and adults, and that the extra genes on chromosome 21 are really involved in many key metabolic pathways, including cholesterol, choline, purine, and reactive oxygen species. And so we are already noticing and noting variation in drug metabolism in individuals with Down syndrome, especially as it relates to methotrexate, because there was toxicity that was related to patients getting the same dose as typically developing children. We also know that there are variations in utilization of, say, acetaminophen, theophylline, donepezil, aspirin, and many of the drugs that are used to treat attention deficit disorder, which is fairly common in individuals with Down syndrome. So just taking methotrexate as an example, individuals with Down syndrome who take methotrexate or who are prescribed methotrexate can experience more cardiotoxicities, a greater rate of infection, mucositis, and actually a suppression of the bone marrow. And so we have to understand this, and there could be several reasons for it which are being studied. One is because of the increased sensitivity due to alterations in polyglutamation of methotrexate. And remember that one of the genes for folate metabolism is actually located on chromosome 21. And when blood levels are taken, there are increased serum levels of methotrexate in individuals with Down syndrome versus controls. So we've just hit now here the tip of the iceberg, but there's much more that needs to be studied as it can really impact safety and also deliverability in terms of efficacy for our individuals with Down syndrome. Now let's take another example of something even like malignancy, how we cannot extrapolate from the general population. We all know that leukemias, both acute lymphocytic leukemia and myeloid leukemias, are much more frequent in individuals with Down syndrome. But I think the other aspect that we are now learning is that the solid tumor risk is actually decreased, except for testicular cancer risk. And once again, the genes on chromosome 21 and the gene dosage is likely playing a role here. So how does this all impact as we're trying to move the field forward? And I'll put this in perspective in terms of clinical trials to look for mechanisms to enhance cognition in Down syndrome. I think we face challenges right from study design to test battery to what's the medication dose, the frequency of the dose, what kinds of individuals can be recruited, consent versus assent, how do we establish safety in a group of individuals where there are challenges in them able to communicate if they're experiencing a side effect, and then funding opportunities. So when we're looking at measures for cognition, I think we can have direct measurements, which are one-on-one interactions with the individuals or by using computer devices. And then there are indirect measurements in terms of behavioral observation. And I think as you'll see from this slide, we have lots of challenges. The skill set of the individual, their ability to pay attention, the time of the day when the test was performed, the test measure, whether it's hitting a floor effect or a ceiling effect or a learning effect. And sometimes we use tools which are also challenging for them, for example, which requires fine motor capabilities. So as test batteries are being assembled, we're trying to look at several different domains, which will capture language, mood, attention, adaptive functioning, quality of life, mental status, etc. But a challenge that still remains in this field, right from the journey where we had begun as early as 2006, is we still don't have a generally accepted test battery to monitor certain medication effects in the context of cognition for individuals with Down syndrome. So advances have been made. An example is the Arizona Cognitive Test Battery. Another example is use of the TestAD battery, which is used in comparison with age-matched, typically developing adults, in areas and domains like attention, psychomotor speed, etc. And that we are also recognizing that language and executive function deficits, these are predictors of functional change. So are we capturing these domains well for individuals with Down syndrome? This is a study which was a group effort, again, looking for cognitive scales, examining memory and executive function, and trying to just even look for suitability and reliability of existing scales in the sense of test-retest reliability. And these were the measures that were looked at. And there were some nice conclusions that were drawn, that there are certain measures that we have been using which would not be appropriate for testing cognition in individuals with with a Down syndrome. And so, yes, the field is progressing forward, but we've learned a lot and we've got a lot to keep in mind, that individuals with Down syndrome can have a very broad spectrum of phenotype. They have significant delays in expressive language when you compare them to to other individuals with a disability. Clinical trials are time limited, so can you really capture the impact of a drug in a short span of time? We need appropriate cognitive measures. And so improvement in cognitive function, which is likely a gradual process, not a dramatic shift. Can we capture it within a clinical trial? How do we ensure that treatments can continue over time and that we continue to understand drug metabolism and drug dosage in individuals with Down syndrome? We're only taking the tip of the iceberg when we enroll patients in these cognitive trials, and then there are these extreme phenotypes, too high functioning or too low functioning. How do we really help those individuals out? Environmental factors and interventions are still necessary. Better understanding of drug metabolism in terms of PK/PD studies are needed, both to test safety and ensure safety and efficacy. A refinement of tools to capture change, better neurological and neurochemical effects of pharmaceutical therapy. We don't have appropriate biomarkers. We are starting to learn much more in terms of neuroimaging, using MRI, MRS, CT, MR or nuclear cerebral perfusion imaging. Some of these are fairly involved procedures, and individuals with Down syndrome, it's a challenge to get them to do these kinds of measures. There is pharmaceutical interest in drug development, but we do need the help of groups like the Pediatric Trial Network to help us understand better impact of drug metabolism so that we can do other studies to understand our individuals with Down syndrome. And then I would really like to close that we must take more than the approach of treating even chromosome 21. We need to have a genomic approach to understanding the spectrum of Down syndrome. And with that, I would like to end and give my strong thanks to our research and clinical team at Duke, to our multiple collaborators, not just in the US, but throughout the world, the Anna's Angels Foundation, and the NIH. Thank you so much.
All right, good morning, everyone. Thank you so much. I'm Mara Becker. I'll be moderating this first session with the speakers from our first session. I'd like to first of all thank, of course, Dr. Parisi, Dr. Zimmerman, and Dr. Kishnani for participating this morning and giving us a great kickoff for the day. I was going to start off with a couple of questions as I review the chat. Please feel free to send more questions into them. I was going to start with Dr. Kishnani and ask in your broad experience, which is clear during your talk, in certainly treating children and conducting trials and studies in children with Down syndrome, what are some of the greatest challenges that you have faced, and how have been some strategies that you've employed to overcome some of those challenges?
Thank you so much, and good morning, Mara and everyone here. I think one of the biggest challenges has been, as I even described in my talk earlier, is how do we really evaluate our children with Down syndrome? So really, do we still have a battery after all these years of studying and evaluating our children? And I would say the answer is no. The second is we really have been in a good space now with the Include project, with help from Melissa and the NIH in spearheading our cause. But now it is more all of us working together to move the field forward. And then, the focus of the PTN, which I think is so terrific, so much needed. Even as we embarked on our clinical trials, we were taking drug doses from, approved package labels and trying to use it in our children with Down syndrome or adults with Down syndrome. And that was a huge limitation. And then I would say is how do we get our individuals and families to be committed, because it does require also caregiver time, follow-up time, and trying to do things in a way where we can limit what we are asking of them, like blood draws, keeping the order straight, and really working closely with experts and learning from others as we propel the field forward. We have come a long ways from where we were, and we are just hoping for much more.
I think that's a great answer, Dr. Kishnani. I appreciate that. And I particularly think you totally queued up our other two speakers here for a second. And so I'm going to tap on Dr. Parisi, since you talked a little bit about Include, and I know particularly some of the amazing work that you've been doing to bring together scientific workshops and people and all of the stakeholders that on the science side of things, as well as patient or advocate and self-advocate stakeholders. Tell me a little bit about what you've learned from that and how that's been shaping the future direction of the program.
Well, it's been really a pleasure to work with the entire Down syndrome community on the Include project. And, we've been able to use some of the scientific workshops to help us design some of the funding opportunity announcements, to help us shape research priorities, and they're actually helping us inform the revisions to the research plan, including the Down syndrome research plan. In addition, it's been really fun because the workshop that we hosted in September of 2019 directly resulted in bringing some investigators together who hadn't known each other before, and they've been now partnering together and submitting grants together that we think are going to advance the field. And of course, bringing in the input from the broader Down syndrome community to inform research has been really instrumental. And having families, self-advocates, and many others who have a stake in the success of Down syndrome research inform and give us feedback about the project at every step of the way has been really essential.
Awesome. In our last minute, Dr. Zimmerman, tell us what the PTN is so spectacular at. I'd love to hear a little bit about what the process is that PTN is so good at that helps us get this kind of program off the ground.
Absolutely. Thank you very much, Mira. So I think the PTN, as I mentioned, and as Dr. Kishnani mentioned before, she set me up very nicely, actually. Some of our expertise is really in the PK and PD. So how do we find the right dose for patients? We've done investigations in patients who are obese, in children who have other genetic type syndromes. So we're really very well set up in order to identify what the right dose is for this particular population. The second thing is about infrastructure, identifying and allowing us to use our infrastructure so that we can teach other people how to do this type of work. And then thirdly is our connection with the regulatory bodies and the fact that we really want to do things that move the field forward. We want to make sure that we are finding the correct dose, finding the safe and efficacious dose, but making sure that that can be in the package label. So Dr. Kishnani, when you're treating your patients, you know, as well as the person across the country knows what dose is the appropriate one.
Thank you guys so much. I think we are ready, but I was going to ask Dr. Parisi if you knew in your portfolio of any of the funded research. There was a question in the chat about environmental factors and if there are any applications that you're aware of that are testing that at this point. As far, I think of that as epigenetics, but I think it can go much broader than that. And we can think about that as well and try to answer that along the way.
I could just say one thing really quickly. We are interested in learning about some of the social determinants of health and other factors that can influence risk to develop Alzheimer's disease in adults with Down syndrome. So we're actually funding a project that's looking at aspects such as diet and exercise, social interactions, and how those influence risks of developing dementia later in life. So I think some of the research is actually starting to look at some of those environmental factors that influence development of some of the co-occurring conditions in Down syndrome.
Wonderful. Thank you so much. So I think we are going to be ready to close up this session and move on to the second session, which is the community engagement and readiness section, which I'm so excited about. We're going to be having a number of partners of our self-advocates and their families, as well as some of the community advocacy programs. We're going to end that with an amazing panel, which will be hosted by Maria Stanley from Wisconsin, and we hope that you guys stay tuned. Thanks very much.